Hello everyone. Now in this lecture, I will be talking about the screening of antibiotic producing organism by Wilkins method. If you remember my previous lecture, it was on screening of antibiotic producing organism by a crowded plate technique. At the end of that lecture, I have discussed regarding the disadvantages of crowded plate technique, where I mentioned it was not possible to distinguish whether the inhibition zone around the colony it is because of antibiotic produced by the organism or it is because of acid produced by the organism. So that problem is overcomes by Wilkins method. Now let's see in detail about the Wilkins method for primary screening of antibiotic producing organism. Here we are going to consider soil as a source of organism. Okay, so do the proper dilution of soil. This is just to reduce the load of organism. Then select the particular dilution of soil sample and take 0.1 ml of diluted soil sample and spread it on the agar media. Now here the media which we are going to use, that is called as the Wilkins media, and this contains the special characteristics of this media is that it contains the pH indicator, that is bromothymol blue. Now this bromothymol blue, it is green at pH 7 whereas yellow at acidic pH. Okay, now after spreading that particular dilution of soil sample on this Wilkins media, you incubate those plates at room temperature for 24 hours. After incubation, select the plates having colonies around 300 to 400. Then just observe the colonies. So here we can very well differentiate between antibiotic producer and acid producing organism. Now how can we distinguish over between these two, antibiotic producer and acid producer? See, if you can find out the isolated colony with zone of inhibition of other bacteria without change in the color of media, then we consider it as this colony is antibiotic producer. Or in other case, if we can detect the isolated colony with the zone of inhibition of other bacteria with change in the color of the media, that is from green to yellow. Why there is change in the color? Because this media, as I have mentioned, there is a bromothymol blue, and this bromothymol blue under acidic condition turns yellow. Okay, so if yellow color is there, means if the colony has turned into yellow color and there is an inhibition zone, it means that the colony which is at the center has produced acid, and the inhibition zone is because of acid production. What I mean to tell you that this acid has inhibited the growth of other bacteria. It is not because of antibiotic, and in this case we are interested to isolate and detect the antibiotic producing bacteria, not the acid producing bacteria. Okay, so if you find the change in the color, that is from green to yellow, not only colony, even the color of media get changed from green to yellow. If you find the change in the color of the media, it means that the colony which is at the center of inhibition zone, that is producing acid, not antibiotic. Okay, so ignore this type of colony and go for the colony where there is a inhibition zone but without change in the color. Okay, now subculture, pick up that colony, subculture the selected colony and send it for secondary screening. That will Secondary screening, which will provide us the detailed information regarding the selected organism, as well as the nature of antibiotic, whether it is broad spectrum antibiotic or narrow spectrum antibiotic. Thus this Wilkins method, it helps us to overcome the disadvantage of crowded plate technique. Thank you.